Imagine for a moment if the very cells that make up your body weren't organized, weren't really talking to each other, couldn't even hold their shape properly. Think about how fundamentally different life would be if tissues just sort of unraveled. Sounds almost too basic, right? But the intricate ways cells interact with their environment and with each other are absolutely essential for everything your body does, from, your heart beating to how a simple cut heals. Welcome to the deep dive. We're here to unpack complex ideas and hopefully give you some of those aha moments. Today, we're diving deep into the truly fascinating world of cell and extracellular interactions. We're drawing our insights from a really comprehensive chapter in Karp Cell and Molecular Biology, the 8th edition. Our mission today was to journey through the key concepts, the intricate molecular processes, and some surprising real-world implications of how cells organize, how they communicate, and navigate their incredibly complex world. You're going to leave here not just informed, but I think genuinely amazed by the microscopic dance that's happening inside you right now.
It really is astonishing just how much depends on these tiny, tiny interactions. When we think about the cell's outer boundary, beyond the plasma membrane itself, the first thing we encounter is the glycocalyx.
The glycocalyx, right?
And it's not just random fluff, it's this fuzzy layer of carbohydrate chains, oligosaccharides, projecting outwards. People often call it the cell's sugar coat.
A sugar coat. I like that image.
Whoa.
So, okay, beyond just looking fuzzy, how fundamental is this glycocalyx? What does it actually do?
Oh, it's crucial. It's a multifunctional interface. It mediates key cell and cell substratum interactions, so how cells stick to each other and to surfaces. It offers mechanical protection, acts as a bit of a barrier to particles, and significantly it can bind important regulatory factors. So it's like a docking station for signals that influence what the cell does next. You see this layer very prominently, for example, in the epithelial cells lining our digestive tract. Lots of interaction happening there.
Okay, so definitely more than just fuzz. And moving a bit further out, we hit something more substantial. The extracellular matrix, or ECM. This isn't just biological glue, is it?
Absolutely not. The ECM is this really organized network of molecules that cells secrete. It provides the essential scaffolding for cells and tissues. But it's anything but inert. It's incredibly active.
Active how?
It provides critical signals, physical ones, biochemical, mechanical signals that profoundly regulate cell shape and activities.
Right.
There's a great experiment, for instance, if you take cartilage cells in culture and digest their ECM away with enzymes.
Okay.
Their synthetic activity, what they make and secrete, just plummets. But then if you add the ECM components back, their differentiated state, their specialized function is restored. It really shows that ECM directly influences what a cell is and does.
Wow, okay, so it's actively telling the cells what to be. And within this ECM, there's a specialized layer, a sheet, called the basement membrane. Where do we find these?
Yeah, basement membranes are critical foundations. They are these continuous sheets, pretty thin actually, maybe 50 to 200 nanometers thick.
Tiny.
Very. And you find them surrounding nerve fibers, muscle cells, fat cells. They also underlie the basal surface of epithelial tissues, think the epidermis of your skin, or the lining of your digestive and respiratory tracts. And really importantly, they underlie the inner endothelial lining of blood vessels.
So they're basically lining and wrapping all sorts of vital structures. Given how widespread they are, what are their main jobs?
They do a lot. They provide essential mechanical support for the cells attached to them. They generate signals that actually help cells survive. They act as a substratum, a sort of highway for cell migration during development or wound healing.
Right.
They also physically separate adjacent tissues within an organ, and they function as a selective barrier controlling what large molecules can pass through. A key example is in your kidneys.
The filtering units.
Exactly. The basement membranes of the kidney capillaries are vital for filtration. They stop proteins from your blood leaking out into the tissues. And when this goes wrong, the consequences are serious. Like in long-term diabetes, abnormal thickening of these basement membranes can lead to kidney failure. They also act as a barrier against cancer cells trying to invade tissues.
It's incredible how critical these thin sheets are. So what are they actually made of? What are the building blocks giving the whole ECM its strength and flexibility? Let's start with the big one, collagen.
Right, collagen. It's the most abundant protein in mammals, actually, made primarily by fibroblasts in connective tissues, but also by smooth muscle and epithelial cells.
And there isn't just one type, is there?
No, far from it. There are at least 28 distinct types identified in humans, and each type tends to be restricted to specific locations in the body. What's really neat is how different types are often mixed together to create what we call heterotypic fibers.
Like alloys.
Exactly like biological metal alloys, it allows for varied structural and mechanical properties, perfectly tailored to what that specific tissue needs.
That's clever. But they all share a core structure.
They do. All collagen molecules are trimers. That means they're made of 3 polypeptide chains called alpha chains, wound into this characteristic rod-like triple helix.
Okay, the triple helix.
And a crucial detail for stability is their high content of proline and lysine. These amino acids get hydroxylated and OH group is added after the protein chain is made. This hydroxylation is absolutely vital because it allows hydrogen bonds to form between the chains, stabilizing that helix.
And this is where vitamin C comes in, right? Scurvy?
Precisely. Scurvy, caused by vitamin C deficiency, has such devastating effects: inflamed gums, lost teeth, poor wound healing, brittle bones, weak blood vessels, because vitamin C is essential for the enzymes that do that hydroxylation, unstable collagen, weak tissues.
Wow, that really connects the molecular detail to the systemic effect. So structure-wise, you mentioned fibrillar versus non-fibrillar collagen.
Yes. The fibrillar collagens, like types 1, 2, and 3, assemble into these very rigid, cable-like fibrils, which then bundle into thicker fibers. They provide immense tensile strength resistance to pulling forces. Think of them as the body's heavy-duty ropes.
Okay.
The cross-linking between these fibrils increases as we age, which contributes to things like skin losing elasticity and bones becoming more brittle.
And the non-fibrillar.
Non-fibrillar collagen, like type 4, is mainly found in basement membranes. It's more flexible, it has these globular ends, and forms more of an irregular lattice network, not those rigid cables. Perfect for the flexible sheet-like structure of a basement membrane.
The properties of tissues really show this off, like tendons need that rope-like strength.
Exactly. In tendons, the collagen fibrils are all aligned parallel to the direction of pull, giving them incredible strength. But then look at the cornea of your eye.
Needs to be strong, but clear.
Right. Its thick middle layer, the stroma, has short collagen fibrils organized in layers, almost like plywood, with each layer perpendicular to the next. This gives strength, but the really uniform size and precise packing of the fibrils minimizes light scattering. That's why it's transparent.
It's just amazing architecture at the micro level.
Well-
But, of course, things can go wrong. What happens when collagen formation is faulty?
Yeah, abnormal collagen leads to some serious conditions. Fibrosis, for instance, is basically excessive collagen production. It can replace normal tissue in organs like the lungs or liver that leads to pulmonary fibrosis or cirrhosis.
Okay.
Then you have genetic disorders. Mutations in type I collagen cause osteogenesis imperfecta or brittle bone disease, extremely fragile bones. Type 2 mutations can lead to dwarfism and skeletal issues. Other mutations cause Ehlers-Danlos syndromes with hyperflexible joints and stretchy skin. And mutations in type 4, the basement membrane type, cause Alport syndrome and inherited kidney disease because the glomerular basement membrane is disrupted.
This highlights how fundamental collagen is. Okay, moving on. The next component sounds interesting. Proteoglycans. You called them hydrated gel fillers.
That's a great way to think about them. They are these very large complexes of protein and polysaccharide. Structurally, you have a core protein, and attached to it are these long chains called glycosaminoglycans, or GAGs.
GAGs. Yeah.
These GAG chains are repeating disaccharides, and they're highly acidic because they have lots of sulfate and carboxyl groups. These proteoglycans can get enormous, especially when they link up to another molecule called hyaluronic acid, forming huge complexes.
And their main function is cushioning.
Yeah.
Resisting compression.
Precisely. Because those GAGs are so acidic, they bind huge numbers of positive ions, cations, and these cations in turn bind massive amounts of water. This creates this porous, hydrated gel that fills the extracellular space.
Ah, like a sponge.
Kind of, yeah. It resists crushing forces incredibly well, so it perfectly complements collagen, which resists pulling forces. You find lots of proteoglycans in cartilage and bone, giving them their resilience. You even see JAGs like chondroitin sulfate sold as supplements for joints and skin.
Right, I've seen those. Do they
Yeah.
do anything else?
They do have signaling roles, too. They can bind growth factors like FGF and VEGF. They might act as a reservoir or help form stable gradients of these signals, guiding cell behavior.
So definitely not just passive filler. Next up, fibronectin. This one was described as a guiding network.
Fibronectin is fascinating. It's a modular protein built from about 30 distinct structural units called N-domains. What's really interesting is that these domain types are found in lots of other proteins too, suggesting they were maybe mixed and matched through evolution by gene fusion.
Modular design. And it connects things.
Yes, it has key binding sites for other ECM components, collagens, proteoglycans, and also for cell surface receptors, specifically integrins, which we'll get to. So it forms this crucial bridge, linking the ECM network directly to the cell.
And I heard something about it being dynamic, like it can change shape.
That's right. Mechanical forces, like the cell pulling on it, can actually unfold some of these modules. This can expose hidden binding sites, potentially changing its interactions or signaling properties.
Wow.
This dynamic nature is vital, especially in development. Fibronectin plays a huge role in guiding cell migration. Neural crest cells, for example, follow along fibronectin-rich pathways as they migrate through the embryo. It's also essential for how organs like salivary glands, kidneys, and lungs develop their branching structures. If you block fibronectin function with antibodies, that branching process just stops.
Like a cellular roadmap.
Hmm.
Okay, and the last major ECM component we'll cover is laminin, the cross-shaped integrator.
Laminins are another family of extracellular glycoproteins. They have this very distinctive cross-like shape made of 3 different polypeptide chains wound together. There are at least 15 different types known.
And their role, similar guiding function.
Yes, they critically influence cell migration, growth, and differentiation. For instance, the primordial germ cells, the precursors to sperm and eggs, migrate from the yolk sac to the developing gonad along surfaces rich in laminin.
So another key player in guiding cells.
Definitely. Laminins bind to cell surface receptors. They bind to each other, to proteoglycans, and importantly, to other basement membrane components like type V collagen. They form these intricate, interwoven networks with type V collagen, giving basement membranes both their strength and their flexibility. Their importance is underscored by knockout studies. Mouse embryos that can't make laminin fail to form any basement membranes and die very early in development. Absolutely essential.
Okay, so we have this incredibly complex and dynamic environment outside the cell, the ECM, the basement membrane, built from collagen, proteoglycans, fibronectin, laminin. How do cells actually sense and talk back to this environment? This brings us to integrins, the cell's dynamic communicators.
Exactly. Integrins are the key players here. They are integral membrane proteins, meaning they span the cell membrane, linking the cell's interior cytoskeleton to the exterior world, the ECM.
And structurally?
They're made of 2 subunits, an alpha and a beta chain. Together, they form a globular head that sticks out of the cell, transmembrane legs that go through the membrane, and a small cytoplasmic domain inside the cell. What's really cool is they can switch between 2 main shapes: a bent, sort of low-affinity inactive form, and an upright high-affinity active form. They're like molecular switches.
Perfect for signaling. You mentioned inside-out and outside-in signaling. Can you break that down?
Sure. Inside-out signaling is when something inside the cell triggers the integrin to change shape. For example, a protein called talin might bind to the integrin's cytoplasmic tail. This causes the integrin to straighten up, become active, and bind more strongly to its ligand outside the cell. This often leads to integrins clustering together, making adhesion much stronger.
Okay, so the inside tells the outside to grab on tighter.
Right. Then there's outside-in signaling. This is when something outside the cell, like fibronectin or collagen in the ECM, binds to the integrin's external head. This binding causes conformational changes that propagate inside the cell.
And what does that trigger inside?
A whole range of things. It can trigger the polymerization of actin filaments, reorganizing the cell's cytoskeleton. It can activate protein kinases inside the cell, like FAK and SRC, which are key signaling hubs. It can even lead to changes in gene expression way down in the nucleus.
They really are two-way streets,
Yeah.
influencing so much.
Yeah.
Differentiation, movement, even survival.
Absolutely. Integrin signals have a profound impact on cell differentiation, motility, growth, and yes, survival. It's striking that most normal cells actually need to be anchored via integrins to survive. If you put them in suspension where they can't attach, they often undergo apoptosis, programmed cell death. Malignant cells, however, frequently lose this dependency. That's one reason they can detach and spread. And a key molecular detail. Many integrin ligands, the things they bind to in the ECM, contain a specific 3 amino acid sequence: arginine, glycine, aspartic acid, RGD.
RGD. And knowing that sequence has medical applications, like with blood clotting.
Yes, definitely. Platelets, the little cell fragments involved in blood clotting, use a specific integrin, alpha I beta 3, to bind to blood proteins like fibrinogen that contain that RGD sequence. This binding creates bridges between platelets, holding them together to form a clot and seal injuries.
So understanding that RGD interaction led to drugs.
Exactly. Researchers developed synthetic RGD peptides that can compete with fibrinogen for binding to the platelet integrin. This inhibits clot formation. That led directly to antithrombotic drugs like aggrastat and integrelin. There are also therapeutic antibodies that target specific integrins like RioPro, used during certain heart procedures, or Tysabri, used to treat multiple sclerosis by blocking inflammatory cells from crossing the blood-brain barrier.
That's a fantastic example of bench-to-bedside translation. Do different integrins have unique roles, even if they bind similar things?
They certainly do. Experiments using knockout mice where specific integrin subunit genes are deleted show this clearly. Mice lacking different subunits have very distinct problems. Kidney defects, heart defects, vascular problems, highlighting that each integrin pair has crucial, non-redundant functions.
Okay, let's shift focus slightly to how cells physically anchor themselves. When a cell lands on a surface, it doesn't just sit there, right? It spreads out.
Right. It transitions from a rounded shape to flattening and spreading, sending out projections, and it forms specific attachment points.
And those are the focal adhesions, dynamic contact points.
Exactly. Focal adhesions are these discrete scattered sites where the cell anchors itself firmly to the substratum, the surface it's on, but they're highly dynamic. They can assemble and disassemble quite rapidly, which is essential if the cell needs to move or divide.
What's the structure like at these points?
At a focal adhesion, you have large clusters of integrins packed together in the plasma membrane. Their cytoplasmic tails are linked via a whole network of adapter proteins like taurine, alpha-actinin, and vinculin to the actin filaments of the cytoskeleton. So they physically connect the outside world to the cell's internal framework.
And they're not just passive anchors, they're involved in sensing forces, this mechanotransduction.
Yes, this is a really hot area of research. Focal adhesions act as mechanosensors. They can literally sense the physical properties of the environment, like stiffness or tension, and convert those physical cues into biochemical signals inside the cell.
How precise can this sensing be?
Remarkably precise. There are experiments using techniques like optical tweezers to pull on individual integrin molecules. They found that strong focal adhesions would only form if the integrin was linked to a molecule that could withstand a certain amount of force, say 56 piconewtons, but not if it was linked to something weaker, like 43 piconewtons. It suggests cells can measure forces at the molecular level.
That's incredible. And this sensing affects cell fate, like stem cell differentiation.
It does, profoundly. Studies have shown that the stiffness of the substrate a stem cell is grown on can actually direct its differentiation pathway. Put mesenchymal stem cells on a soft gel, similar to brain tissue, and they tend to become nerve cells. On a medium stiffness gel, like muscle, they become muscle cells. And on a stiff surface, like bone, they differentiate into bone-forming osteoblasts.
So the physical feel of the environment is telling the cell what to become. That's amazing. Now, for really strong adhesion, especially for epithelial cells anchoring to the basement membrane, there's another structure, hemidesmosomes.
Right. Hemidesmosomes are arguably the tightest adhesive contact cells make with the extracellular matrix. They're crucial for holding epithelial layers, like your skin, firmly onto the underlying tissue.
How are they structured? Similar to focal adhesions.
They share some features, but are distinct. They have a dense plaque on the inner surface of the plasma membrane. But instead of linking to actin filaments, they anchor intermediate filaments, specifically keratin filaments in epithelial cells, which loop through the plaque and extend deep into the cytoplasm.
Intermediate filaments, not actin.
Exactly. And the integrin involved is typically alpha-6 beta-4. This integrin connects the keratin filaments inside, through the plaque and membrane, to specific components in the basement membrane outside, particularly laminin 5 anchoring filaments and type 7 collagen fibrils. It's a multi-part connection providing really robust mechanical stability.
Super strong anchors. And the real-world relevance comes through again with blistering diseases.
Tragically, yes. Bullous pemphigoid is an autoimmune disease where the patient's own immune system makes antibodies against proteins in the hemidesmosome. This disrupts the connection, causing the epidermis to literally detach from the underlying dermis, resulting in severe blistering. And epidermolysis bullosa covers a group of inherited blistering diseases caused by genetic mutations in genes encoding hemidesmosomal proteins, could be the integrin subunits, collagen 7 or laminin 5. Any weakness in that chain leads to fragile skin that blisters easily.
Really underscores their importance for tissue integrity. Okay, let's broaden the view again. We've talked cell to ECM, now let's focus on cell-to-cell interactions. There's a remarkable specificity here, isn't there, especially in
It's
development?
absolutely remarkable. A classic experiment involves taking embryonic tissues, say ectoderm and mesoderm, dissociating them into single cells and then mixing them together. Initially, they just form random clumps,
Uh,
but then over time, they sort themselves out. The ectoderm cells find and adhere to other ectoderm cells, and mesoderm cells stick to mesoderm cells, eventually reforming segregated tissues. This precision relies on specific cell adhesion molecules, or CAMs.
and there are four major families of these camps. First up, selectins.
Selectins are involved in mediating relatively weak, transient adhesions. Their main job is in the bloodstream, helping circulating white blood cells, leukocytes, interact with the walls of blood vessels, especially at sites of inflammation or clotting. They are crucial for the initial rolling of leukocytes along the vessel wall before they stick more firmly and exit into the tissue.
Transient adhesions, okay.
Hmm.
And what's unique about them?
One interesting thing is that their bonds can actually strengthen under mechanical stress, under flow. As the blood flow pulls on the rolling leukocyte, the selectin bond gets stronger for a moment, which helps slow the cell down.
Clever design. Next family,
Hmm.
the immunoglobulin superfamily, IGSF. Sounds like they're related to antibodies.
They are, structurally. They contain domains called Ig domains, which are similar to those found in antibodies. This suggests they might have evolved from an ancient cell adhesion system even before the sophisticated immune system we know today developed.
So not all are involved in immunity.
No. While many IGSF CAMs are crucial for immune cell interactions, others, like VCAM, NCM, and L1, are really important for mediating calcium-independent adhesion between non-immune cells. NCM and L1, for instance, play big roles in nervous system development, guiding nerve outgrowth.
Okay. And integrins pop up again here. I thought they were mainly cell ECM.
Mostly, yes, but a few integrins do mediate cell adhesion. A key example is the integrin alpha-beta-2 found on leukocytes. It binds to an IGSF molecule called ICAM1 on the surface of endothelial cells lining blood vessels. This is a critical step in inflammation, allowing leukocytes to adhere firmly before migrating out.
Got it. And the fourth, and arguably most important family for holding tissues together, cadherins, the cohesive force.
That's a perfect description. Cadherins are a large family of glycoproteins, and they mediate calcium-dependent cell adhesion. They also transmit signals across the membrane.
Calcium dependent, that's key.
Yes, very key. And their defining property is that they typically mediate homophilic adhesion, meaning a cadherin molecule on one cell binds preferentially to the same type of cadherin molecule on an adjacent cell. This is largely responsible for that sorting out phenomenon we see with embryonic cells. They are arguably the single most important factor in molding cohesive tissues during development and then holding those tissues together throughout life.
How do they work structurally? How do they achieve that strong, specific binding?
They're modular proteins, typically with 5 tandem domains sticking out from the cell surface. Calcium ions bind between these domains, keeping the molecule rigid and extended, which is necessary for adhesion. Without calcium, they become floppy and don't stick well. Inside the cell, their cytoplasmic domain links up to the actin cytoskeleton via a set of adapter proteins called catenins. This connection is crucial, not just for adhesion strength, but also for signaling. When cadherins from opposing cells interact, they often cluster together, forming structures sometimes described as a cell adhesion zipper or acting like molecular Velcro, dramatically increasing the overall strength of the connection.
And this ability to connect or disconnect is vital in development, like in EMT.
Exactly. Epithelial mesenchymal transition, EMT, is a fundamental process in development. Think about gastrulation, where germ layers are formed, or neural tube formation. Cells in an epithelial sheet need to break away, lose their tight cell-cell contacts, become migratory mesenchymal cells, and move to new locations.
And that involves changes in cadherins.
Critically, yes. Often, cells undergoing EMT downregulate their expression of E-cadherin, the main cadherin in epithelia. Losing E-cadherin allows them to detach and become mobile.
And the flip side of that is cancer.
Unfortunately, yes. The loss of E-cadherin expression is very strongly linked to EMT occurring in cancer progression. It allows tumor cells to detach from the primary mass, invade surrounding tissues, and metastasize to distant sites. It's a major factor in cancer becoming invasive and spreading.
So restoring E-cadherin could potentially stop metastasis.
In some experimental models, forcing cancer cells that have lost E-cadherin to express it again can actually reduce their invasiveness and ability to form tumors. There are even inherited forms of stomach cancer linked directly to mutations in the E-cadherin gene.
Wow. Any other connections?
Yes, there are non-classical cadherins involved in sensory perception. Mutations in cadherin 23 or protocadherin 15 cause Usher syndrome, a condition leading to both deafness and progressive vision loss. These specific cadherins form the tip links, tiny filaments connecting the stereocilia on hair cells in the inner ear. These tip links are essential for converting the mechanical force of sound waves into the electrical signals the brain interprets as sound.
Such specific, vital roles. Okay, let's quickly touch on some specialized cell junctions that use these CAMs. First, adherens junctions, the belts of adhesion.
Right. Adherens junctions often form continuous belts called zonulae adherens, encircling epithelial cells, typically just below the tight junctions near the apical surface. They basically connect the actin cytoskeletons of adjacent cells.
And they use cadherins.
Yes. Cadherins bridge the gap between the cells, and their cytoplasmic tails link via catenins to actin filaments. They provide adhesion, connect the outside to the inside, and transmit signals. Endothelial adherens junctions, for example, are known to transmit survival signals to the cells.
Okay. Then desmosomes. These sound stronger spot welds.
They are indeed like spot welds. Desmosomes, or maculae adherens, are disc-shaped adhesive junctions particularly abundant in tissues subjected to significant mechanical stress, like heart muscle or the layers of your skin.
How do they differ from adherens junctions?
They also use cadherins, but specialized types called desmogleins and desmocollins. The big difference is inside the cell. Instead of linking to actin, desmosomes anchor intermediate filaments like keratin. These filaments loop through dense cytoplasmic plaques associated with the desmosome, creating a continuous network of intermediate filaments across the entire epithelial sheet, providing tremendous tensile strength and resistance to shearing forces.
So adherens junctions link actin networks. Desmosomes link intermediate filament networks.
Got it. Now, the seals and fences. Tight junctions.
Tight junctions, or zonulae occludens, are typically found at the very apical edge of the junctional complex between epithelial cells. They form a continuous seal that completely encircles each cell, like the plastic gasket on a food container.
What makes the seal? The seal is formed by networks of transmembrane proteins, primarily occludin and various types of claudins. These proteins on adjacent cells bind directly to each other, forming strands that effectively stitch the membranes together at intermittent points. If you freeze-fracture the membrane, you can see these sealing strands as interconnected ridges and grooves.
And they have two jobs, barrier and fence.
Exactly. The barrier function is to block the free diffusion of water and solutes through the space between the cells, the paracellular pathway. This creates tight seals essential in places like the lining of your intestine to keep gut contents separate from your tissues, or your urinary bladder to prevent urine leakage. The tightness varies depending on the specific claudin proteins present and the number of sealing strands.
And problems here cause disease.
Yes. For instance, mutations in a kidney-specific claudin, claudin 16, disrupt magnesium reabsorption in the kidney tubules because the junction becomes impermeable, leading to low magnesium levels. And mice lacking claudin 1 die from dehydration shortly after birth because their skin isn't watertight.
Wow. And the fence function.
The fence function is about maintaining cell polarity. Tight junctions act like fences within the plane of the membrane itself, preventing integral membrane proteins that belong in the apical domain, facing the lumen or outside, from diffusing laterally into the basolateral domain, facing the underlying tissue and blood, and vice versa. This keeps the different functional regions of the cell surface distinct. They are also critical for forming the blood-brain barrier.
Right, protecting the brain.
Precisely. They seal the endothelial cells of brain capillaries very tightly, restricting the passage of most substances from the blood into the brain tissue. This is protective, but also a challenge for getting drugs into the brain. Researchers are actively trying to find ways to temporarily and safely open these junctions to deliver therapeutics for neurological diseases.
Fascinating challenge. Okay, moving from sealing cells off to connecting them directly. Gap junctions. For communication.
Yes, gap junctions are specialized sites that allow direct cytoplasmic communication between adjacent animal cells. It's like having little pipelines connecting their interiors.
How are they built?
At a gap junction, the plasma membranes of the two cells are very close together, separated by a narrow gap, but they don't fuse. This gap is spanned by channels called connexins. Each connexin in one cell lines up perfectly with a connexin in the adjacent cell to form a continuous aqueous channel. And each connexin itself is made of 6 protein subunits called connexins.
So, like little tunnels, what passes through?
They allow the rapid passage of small molecules and ions up to about 1,000 Daltons in molecular weight. This includes ions, which allows for electrical coupling between cells, as well as small metabolites like ATP, cyclic AMP, amino acids, and sugars. They're relatively non-selective for small molecules, but can be gated, opened, or closed in response to various stimuli like changes in voltage, pH, or calcium levels, or phosphorylation of the connexin proteins.
And where's this direct communication critical?
Extremely important in several tissues. In cardiac muscle, they allow the rapid spread of electrical impulses from pacemaker cells throughout the heart, ensuring coordinated synchronous contraction. Same idea in smooth muscle, coordinating waves of contraction like peristalsis in the gut. They also allow for metabolic cooperation between cells, enabling cells in tissues without direct blood supply, like the lens of the eye, to share nutrients and metabolites. Overall, they integrate the activities of individual cells into a functional unit.
Are there diseases linked to gap junctions?
Yes, inherited mutations in connexin genes are linked to a surprising range of conditions, including certain types of deafness, blindness, cataracts, skin disorders, nerve disorders, and cardiac arrhythmias.
Like cancer, I remember hearing something about that.
There's a really interesting connection. Early studies observed that many types of cancer cells seem to have reduced or absent gap junction intercellular communication, GJIC. What's more compelling is that experiments forcing these cancer cells to express functional connexins and reestablish GJIC can often dramatically slow their growth rate and reduce their ability to form tumors. This suggests that GJIC might normally play a role in suppressing uncontrolled cell growth.
That's a potential therapeutic angle, too.
Yeah.
Are there other ways cells connect over distances? You mentioned tunneling nanotubes.
Yes, this is a relatively newer discovery. These are very thin, membrane-bound tubules, much longer and thinner than gap junctions that can stretch between cells, sometimes over quite long distances. They seem capable of transferring not just small molecules, but even larger cargo like cell surface proteins, vesicles, mitochondria, and possibly even infectious particles like viruses or prions between connected cells. Still a lot to learn about them.
Whole other communication network.
Mm.
And finally, plants. They have cell walls, so how do they manage direct connections? Plasmodesmata.
Exactly. Plasmodesmata are the functional equivalent of gap junctions in plants. They are cytoplasmic channels that pass directly through the rigid cell walls, separating adjacent plant cells, effectively connecting their cytoplasm and allowing for communication and transport.
How are they structured?
Each plasmodesma is lined by an extension of the plasma membrane, and running through its center is a narrow tubule called the desmotubule, which is derived from the endoplasmic reticulum of the connected cells. So they connect both the cytoplasm and the ER networks.
And they allow passage of?
Similar to gap junctions, they allow passage of small solutes, but remarkably, they can also traffic larger molecules, including proteins and RNA molecules. This transport is often regulated by specific plant movement proteins. This allows for complex signaling and coordination throughout the plant, integrating activities like growth, development, and defense against pathogens across different tissues.
So plants have their own sophisticated way of staying connected despite the walls. Speaking of which, let's just briefly touch on plant cell walls themselves as a point of contrast.
Right. While animal cells typically only have the plasma membrane and maybe a glycocalyx or ECM, almost all other organisms, plants, fungi, bacteria, even many protozoa have some kind of protective outer envelope beyond the membrane. The plant cell wall is a classic example, and actually it was one of the very first cellular structures observed historically by Robert Hooke using an early microscope.
What's the main component? Cellulose.
Cellulose is the major structural component, yes. It forms rigid microfibrils that provide tensile strength. These are embedded in a matrix made of other complex polysaccharides like hemicellulose and pectin, which are synthesized in the Golgi apparatus and secreted. There are also structural proteins within the wall. Interestingly, some proteins, like expansins, can actually loosen the wall structure locally, allowing the cell to expand during growth. So, like the animal ECM, the plant cell wall isn't static. It's a dynamic structure that can be modified.
Wow. Okay, we've covered a huge amount of ground. From the cell's sugar coat, the glycocalyx, through the complex scaffolding of the ECM with its collagen cables and proteoglycan cushions, the crucial basement membranes.
to the integrins acting as dynamic communicators linking inside and out, the focal adhesions and hemidesmosomes anchoring cells, the specific cams like cadherins holding tissues together.
And the specialized junctions adherens, desmosomes, tight junctions, gap junctions providing strength, sealing, and direct communication. And even a nod to plant plasmodesmata and cell walls. What really stands out from this deep dive is that cell interactions are so much more than just cells sticking together. It's constant dynamic communication. It's about sensing the environment, responding, coordinating actions, maintaining health, and unfortunately also involved when things go wrong in disease.
It leads to a pretty profound thought, doesn't it? If a single cell's ability to talk to its neighbors and its environment dictates its survival and function, and the combined communication of trillions of cells creates us, creates tissues, organs, entire organisms, what else could we learn? What new insights might we unlock by digging even deeper into these fundamental biological conversations?
Exactly. Think about the potential applications. If we can understand these interactions with even greater precision, could we design therapies that specifically target faulty connections in disease? Imagine guiding tissue regeneration more effectively, or finding new ways to prevent cancer cells from breaking loose and metastasizing, just by manipulating these specific adhesion and signaling molecules.
The possibilities are really exciting.
The more we explore these intricate biological systems, the more appreciation you gain for just how complex and, frankly, elegant life is at this microscopic level. Hopefully this dive sparks some of that curiosity for you. Thank you so much for joining us on this deep dive into cell and extracellular interactions. Until next time, stay curious and keep exploring the incredible world around and definitely within you.